Welcome to today's webinar. This webinar is part of a three-part series on InterPro. And today we are joined by Tiffany Pessin-Lafosse, who is going to talk us through understanding InterPro families, domains, and functions. This webinar will be followed by two others, the next one being next week on using the InterPro website in your research, and the final webinar on the 20th of May on accessing InterPro programmatically.
Hello, everyone. As Melissa mentioned, this webinar will be about understanding InterPro families, domain, and functions. So before I start, what is InterPro? So InterPro provides functional analysis for protein sequences, and to do so, it predicts protein families and domains and important sites on these protein sequences. So how does that work? So let's imagine you have a protein sequence like this one. And InterPro, what does InterPro do is that it assigns, for example, in the full length of the sequence, protein family. But in InterPro, we also assign domains alongside the protein sequence, which are shorter regions in the protein. Finally, we also provide information about important sites like binding sites or conserved sites. So all this information is contained in predictive models from several member databases that are part of the InterPro consortium. So for example, here, if we take this protein family, we can see that it has been predicted by two member databases, the member DB2 and the member DB6. This domain 2 here has been predicted by 3 different member databases. So all this information from these different member databases, which I'm going to talk about in this webinar, are then integrated in InterPro entries, which are represented here by the gray rectangles. And all the information available in InterPro have literature reference based, and this is for the protein families, the domains, and the important sites. So throughout this webinar, I'm first going to give you a few definitions that are important to understand how InterPro works. Then I will present you the different methodology used by the different InterPro member databases. I will also present you a few additional information that we made available, like, for example, disordered regions or per residue annotation. And I will carry on by presenting you the different relationships that we put in place between the different InterPro entries. And I will conclude by presenting you a few resources that are using InterPro as an underlying resource. So let's start with a few definitions about InterPro. So as I was mentioning in the introduction, InterPro does a functional analysis of protein sequences. Using different protein signatures, also called predictive models, from the different member databases. So if we have a protein sequence like this, we can assign either on the full length of the protein, which are protein families. So for example, here, this protein is matching the breast cancer type 2 susceptibility protein family. But smaller region of the protein are also determined, like, for example, the breast cancer type 2 susceptibility protein helical domain, the OB1 domain, or the TOR domain. We also provide structurally based predictions, like, for example, the BRCA2 helical domain superfamily, or the nucleic acid binding OB fold. So all these protein families and domains are present in one or more databases from the member database consortium. For example, the BRCA2 protein family has been predicted by the Panther member database. The helical domain has been predicted by PFAM, and the BRCA2 OB1 has been predicted by the CDD and the PFAM databases, and so on. So InterPro consists of a consortium of 13 member databases, which are Panther, Prints, MobiDB, TigerFAM, CDD, SFLD, ProCite, HAMAP, Superfamily, PFAM, PIR, SMART, and Gen3D. So all these member databases are combined or not, depending on what they are matching, into InterPro entries. But what is an InterPro entry? So an InterPro entry defines one family, domain, or conserved site. And to do so, we assign the InterPro entry based on the matches to protein signatures that model the entity being described. So from one or more of our InterPro member database consortium, they are integrated in this InterPro entry. This is done based on the protein sets they are matching. Like, for example, here we have different proteins. And all the information provided in the InterPro entry is literature supported. So we give a description and we insert, as you can see here, the reference to the literature. And in InterPro entries, we also provide external links and complementary information, like, for example, the domain architecture, taxonomy, proteomes, structures, interaction, pathway, or genome 3D predictions. And we also provide external links, like, for example, here, the ProCite doc, SCOP database, the CAT database, or the EC links for the enzyme references. So now that we have defined an InterPro entry, the InterPro entries are classified in 5 different entry types, which are the homologous superfamily, which contains proteins that share a common evolutionary origin based on their similarity in their structure. We also have the family entry type, which contains proteins that share a common evolutionary origin based on their function, sequences, or structure. We have the domain entry type, which contain functional, structural, or sequence unit that exist in a variety of biological contexts. We have the repeats entry type, which contain short sequences which are repeated within a protein. And finally, we also have the sites entry type, which contains, for example, active sites, binding sites, or conserved sites. So now we have put the base on what is InterPro, and now I'm going to show you the different methodologies that are used by the different InterPro member databases to predict their models. So they are all based on the multiple sequence alignments, and there is 3 different types of methods that can be used. Either the single motif method, also called patterns, the multiple motif methods, also called fingerprints, and the full alignment methods, which are profiles and hidden Markov models, also called HMMs. About the patterns, it is important to remember that sequence features like binding sites or active sites of enzymes consists of only a few amino acids that are essential for the protein functions. So based on this fact, from a multiple sequence alignment, we are going to look at a particular motif that is repeated between the different sequences aligned. From this motif, we extract the pattern sequences, and out of that, we can generate a regular expression. So in this example here, we can see that it's at the first position, we have an alanine or a cysteine amino acid, followed by any amino acid. Then we have valine. Then we have 4 times any amino acid. And at the last residue, we have either a glutamic acid or everything except a glutamic acid or an aspartic acid. And so the member database use this regular expression to design their pattern signature. This method is used by the ProCite database to predict their models. The second method is a multiple motif approach. So this time, instead of a single motif, multiple motifs are defined alongside the sequence. Again, on the motif are extracted and they are used to build the fingerprint signatures. This is used by the Prints member database. Finally, the 2 methods that are using the full length of the alignment to predict their models are the profiles and HMM. So the profiles from a multiple sequence alignment, for the residues, for each residue, it scores the different amino acids according to their frequency at which they occur in the multiple sequence alignment. From that, the scores are weighted according to the evolutionary distance using a Blossom matrix. So what the Blossom matrix does is assign a score depending on the amino acid electrochemical properties. And for each, it assigns the score between pairs of amino acids. So for example, if you have a substitution between 2 amino acids that are both hydrophobic, then those won't have any impact on the interaction in the protein or in the structure of the protein. But if you swap an amino acid that is hydrophobic to an amino acid that is hydrophilic, then there can be changes in the structure of the protein. This method is useful to identify homologs. It is used by 3 member databases integrated in InterPro, which are CDD, ProCite, and HAMAP databases. Finally, the last method is hidden Markov models. So from a multiple sequence alignment, we are going to look at the amino acid occupancy at each position and encode this. And then we also look at the insertion and deletions. And we are going to try to find the best path alongside the sequence here. They can have 3 states, either maintained, deleted, or inserted. So if we look at this example here, at the first position, we don't have any insertion or deletion. So we'll go from B to here. Then second position, no modification. Third position, no modification. And then after the third position, we can see that there is an insertion. So we are going to go to the insertion path. And after that, there is a deletion. So we go to the deletion path. And then we carry on and go to the last amino acid. And this is how we have modeled our motifs. So all the amino acid probabilities are calculated for each position. This is a good method to model very divergent regions of the alignment, and it is good to identify evolutionary distant homologs. This is the most commonly used method for the InterPro member databases. It is used by the SFLD, PFAM, SMART, TigerFAM, Panther, PIR, Gen3D, and superfamily. Another thing I'd like to mention is the PFAM database. So it is a member of the InterPro consortium, and there is a close relationship between PFAM and InterPro because it is maintained by the same group and has the same curator. So it's a database of protein families and domains. And the last release, which was PFAM 33, there was 18,197 entries. Another interesting notion they are providing is the PFAM clans, which are groups related families that have come from a single evolutionary ancestor. This is useful to provide insights for domains with unknown function, and it is also useful to identify more distantly related structural homologs. So in InterPro, we also provide additional annotation, like for example, the per residue annotation. Well, this is currently provided in the InterProScan sequence search by the CDD and the SFLD databases. So what is the per residue annotation? So for example, we have a protein signature from the CDD, and they have assigned, for example, a particular site alongside the signature that has is the binding site, let's imagine. So what InterProScan does is that for all the signatures that are found matching your protein of interest, it is then going to look if this binding site and this amino acid that was at this position is the same in all the different sequences that are matching. And if it does, then you can filter the results. In the future, we are planning to include further per residue annotation from 5 other member databases. Another interesting feature we are including in the results of our InterProScan search is the intrinsic disordered regions. So those regions are floppy regions with non-conserved amino acid, which allow to give the flexibility of the protein structure. And they are linear motifs, and they are involved in coupled folding and binding. The problem with those regions is they are not very well conserved. So it is hard to model their signatures. There is different predictors that are required to capture complementary aspects about this intrinsic disorder. This is provided by the MGnify database, which has 8 different disorder predictors, and they are defining disordered region if it matches at least 5 of the disorder predictors. So if a region is matching those 5 predictors, then they define it as an intrinsic disordered region. This information is available in the other feature sections in the InterPro website. And for more detailed information, you can also go to the MGnify website. So the addition to this intrinsic disorder information is interesting because it fills the gaps on regions where there isn't any InterPro coverage. So to summarize, In InterPro, there is 13 member databases, which are represented here, and they are using different methods to predict their models, which are the profile HMMs, the profiles, the patterns, and the composition prediction. Those different models are integrated into different InterPro entries, which are the homologous superfamilies, the domain, the families, and features and site, and intrinsic disorder, and also repeats. So now we have put the base of all the InterPro entries, different member databases. So now let's look at how we can add the relationship between the different InterPro entries. So let's first look at the relationship between different protein families. So here you have a tree, a hierarchy of different protein families. So if we look more in detail, at the top here we have the parent, and each arrow is leading to a child. So for example, the J protein coupled receptors protein family is a parent of different children, like for example, the secretin-like GPCRs, the CMP receptors, the rhodopsin-like GPCRs, the metabotropic tropic glutamate receptors. If we take the example of the rhodopsin-like GPCRs, they are the parent of the opsins, APG receptors, relaxin receptors, adenosine receptors, and dopamine receptors. And more specifically, the opsins can be subdivided in different families, which are the red sensitive opsin, the red-green sensitive opsins, the blue sensitive opsins, and the rhodopsins. So what we want to represent in this hierarchy is that the lower you go into the hierarchy, the more specific the protein family is. So let's look at an example. If we take, for example, the protein 28682, it will have the protein properties of all the protein families in the hierarchy. So it will have the properties of the J protein coupled receptors, which are membrane receptors interacting with the G proteins and have a 7TM structures. More specifically, it will have the rhodopsin-like GPCRs more specific property, which are the conserved segments. And then it will have the opsins properties, which have lite receptors and bound to chromophores. And finally, it has a specific absorption spectrum of the chromophore. So how do we represent that in InterPro? So this will be modeled in different InterPro entries, which will be, for example, the rhodopsin-like GPCRs are represented by the InterPro entry 276, and they are the parent of the opsins protein family, which has the identifier 1760, which is a parent of the blue sensitive opsins, which has the identifier 1521. So this was an example of the relationship between different protein families. But we also have a relationship between domains in InterPro. So for example, we have the protein kinase like domain, which is a parent of the protein kinase catalytic domain, itself a parent of the serine kinase catalytic domain and the tyrosine kinase catalytic domain. So in InterPro, we put relationships between entry types, entries of the same type, but not between entries of different types. For example, you can't have in the hierarchy a family being parent of a domain. Another type of relationship we have is the overlapping homologous superfamilies. Because of the origin of the homologous superfamilies, which are contain only proteins from evolutionary origin that have a common structure, they are a particular type. And to not lose the link with other InterPro entries, we have introduced these overlapping homologous superfamilies. They are calculated automatically at each InterPro release by analyzing the overlap between matched sequence sets. To be considered as an overlapping homologous superfamily, the two InterPro entries compared have to obey two different conditions. The first one is that the two InterPro entries have to have protein sequences that are overlapping, which means that they are matching the same region of the protein. And the second condition is that the Jacquard index, which make them the two entries considered as equivalent, or the containment index, which defines a parent-child relation of the matching sequence sets, should be higher than 0.75. So this is everything we have about the different relationships. So we have relationship between entry types, entries that have the same entry types, and we also have the overlapping to homologous superfamilies. One last thing I would like to mention is how we ensure the accuracy in InterPro. So let's imagine the circle represent the protein space. And so, as I mentioned before, in InterPro, we have 13 member databases integrated. Here I have only included 8 of them, but yeah, keep in mind that there is 13. So at each InterPro release cycle, we integrate one or more of the member databases updates, which means we are checking each InterPro entry and the protein set they are matching to ensure that they are still corresponding to what is defined in the InterPro entry. Similarly, at each UniProt update, we also check all the protein space for the verification that it is still matching what is described in the InterPro entry. And finally, we're also doing comprehensive checking and fixing the InterPro entries at each stage of the release process. This can be based, for example, on InterHelp ticket we receive from the users. Okay, finally, let's look at the different resources that are using InterPro as an underlying model. So in InterPro is included the protein sequence search InterProScan, which allows protein and nucleic sequences to be scanned against the different InterPro entries and signatures. So it can be used to search for a single sequence or for less than 30 sequences for the web-based service. But we also provide a downloadable version of InterProScan, which you can install and run locally to process more sequences. Another interesting resource that is included in InterPro entries is a gene ontology. So the gene ontology was, so originally there was different organism communities which were not communicating well between each other. So the gene ontology is a unifying model that has able to put the biological knowledge in a computable format. So all the gene ontology, also called Go terms, are arranged in a hierarchy from less specific concepts to more specific concepts. And they are categorized in three different strands, which are the molecular function, the biological process, and the cellular location. So if we take, for example, the protein kinase, it has the protein phosphorylation biological process. It is found in the cytosol, sorry. So this is a cellular location. And finally, its molecular function is a kinase activity. So the Go term are assigned to gene products, which can be RNA, which are provided by the rFarm and RNA Central database. We can assign to proteins from the UniProt knowledge base, and also it can be assigned to protein complexes, which are provided by the complex portal. These Go terms can be used by the scientists to access gene product functional annotation, to analyze high-throughput genomic or proteomic datasets, to validate experimental techniques, to get a broader view of a proteome, or to obtain functional information of an novel gene product. So in InterPro, as I was mentioning, those Go terms are assigned to the InterPro entries manually based on the scientific literature. So the sequence is searched against the database through InterProScan are also annotated with the Go terms depending on the entry they are matching in InterPro. So let's take an example of a protein sequence that is integrated in InterPro, and it is found in two different InterPro entries. The family, which is a malate dehydrogenase type 1 bacterial, and the lactate malate dehydrogenase C-terminal. So both of these InterPro entries have different Go terms assigned. And to assign this Go term, we really need to be careful because it is then applied to all the proteins that are found matching this InterPro entry and are propagated to the UniProt database, which I'm going to talk about in a second. So the UniProt database uses InterPro and InterProScan to create their UniRule annotation, which is then used to automatically annotate the Trumbull database. They also are using the InterPro and InterProScan to curate the SwissProt database. All the proteins in the UniProt knowledge base are also used by the different InterPro member databases to predict their models. And then it is used again by InterPro to create new InterPro entries. Now, if you want to analyze metagenome, you can use the MGnify resource, where you can analyze microbiome data. So MGnify relies on a lite version of InterPro. So this is the full InterPro sets of member databases, and this is a lite version used by MGnify. It includes only the member database resources that can handle fragmented data and that are fast enough to be feasible for metagenomic data volumes. So this includes the CAS Gen3D, PFAM, TigerFAM, Prints, and ProSite patterns databases. So for example, if you have a metagenome, you can use InterPro. Using InterPro, it assigns about 1/3 of the unknown metagenome using the indifferent InterPro entries. So one last resource I'd like to mention that is using InterPro is the genome properties. So genome properties provides a high level of functional annotation. So how does that work? So let's imagine you have a bacterial genome here and a pathway, for example, the proline biosynthesis from glutamate. So what genome property does is that for each enzyme encoding the pathway, it is going to look if it is encoded by a gene in your bacterial genome. And if it is, then this defines the genome property, and the property is respected. And we can say that this genome is encoding this pathway. This is based on the InterPro entry accession and information. So how is that useful? So for example, here is what you can find in the genome property website. On the left side, we have different genome properties, and on the top here, different organisms. The gray square represent cases where the pathway or genome properties is not found. The light blue are cases where it is partially found, and the dark blue are cases where the genome properties is found. So let's imagine you have a proteome of interest, but you don't know to which species it belongs to. You can then compare, depending on the different genome properties of your genome of interest, to other organisms which have already been qualified. And you can see in this case that your proteome of interest is matching the Chinella species. Okay, let's summarize what we've learned through this webinar. So InterPro combines multiple protein models from 13 member databases into a single searchable resource. All those models are integrated in 5 different InterPro entry types, which are the homologous superfamily, family, domain, repeats, and sites. InterPro can also be used to identify domains and sites and novel sequences, or to define its protein families. InterPro is used in a large-scale annotation of proteins, proteomes, or metagenomes, and it is heavily curated, which allows to identify and correct errors regularly. To finish, I'd like to thank the InterPro team of curators and developers, which are maintaining the InterPro database and websites. And next week, I will run a webinar on how you can use the InterPro website in your research. With that, I'd like to thank you all for your attention. And if you have any question, I'd be happy to answer them.
So there were a couple of questions around statistics. In particular, do you take into consideration the e-values of the member databases before you create an InterPro entry? And if those statistics are looking bad, is an entry still created?
So we don't look at their e-value, but we're looking in the protein space on the protein they are the set of protein they are matching to create the different InterPro entries. So if we see that the set of proteins they are matching is too broad or doesn't correspond to what we're expecting, we are not integrating into InterPro entries. That's the reason why on the InterPro website, at each release, on the release page, which I will explain a bit more next week, we have the total number of models from the member databases and the percentage of integrated signatures from those member databases. So we don't integrate everything. We really are careful to integrate only the models that are relevant.
Other questions. So there was also one about using genomic sequences instead of protein sequences in the analysis. Is that something that you can do?
Not in the website, we don't provide that, but you could run InterProScan using DNA, so you could actually get some, but it gets translated as part of the process. So it actually gets executed on the protein domain, but the user could input DNA sequence. Suppose I want to annotate 500 genes at the same time. Is there any programming way to do that with InterPro? And the answer is yes, we provide a tool called InterProScan.
So there's actually a question about the genome properties as well. And I know that your team works on that, so I will ask the question here. And the question is genome properties used for microbial organisms only?
So it's not only for microbials. The genome properties website actually includes a couple of eukaryotes as part of the pre-calculated. Technically, you could use any genome to run and then try to discover the properties that are present or not in it. We just have run mostly microbial organisms as our starting test, or well, as our starting group of organisms, but any organism can be processed in there. So yes, when you see the taxonomy in the genome properties, you're going to find way more microbial organisms than eukaryotic, but there are a couple of eukaryotic in there.
If you have a question about installation, or if you need help in general, who should people get in contact with?
So you can do so using the contact section in the InterPro website, and the person who is responsible and can answer your question will answer it.
In a general sense, when annotating large numbers of sequence, is there a member database annotation one could rely on more as a general overview ontology of the proteins? For example, PFAM versus superfamily.
The different member databases are using different models, and so the different models they are predicting are complementary in some sense, and that's why we have creating InterPro as well, because we can then regroup the ones that are matching the same set of proteins and are defining the same functionalities. Yeah, so I would say no, there isn't one that is better than the other.
It's more the combination of all of them that gives you the best value.
Yes.
Okay. And another question is, are there workflow pipelines on gene annotation for beginners to have a look at? I'm not sure if you can answer that question.
No, in InterPro we don't provide gene annotation, we provide protein annotation. But I guess on the EBI training platform you can find something about that.
Yeah, so the team that looks mostly at gene annotation at EMBL-EBI would be Ensemble. I think they may have a webinar, or they may have some training materials about that as well. But you can also get in touch with the Ensemble help desk and ask them for advice on how you would approach gene annotation as well. They have a whole team that's really very friendly and can help you out with that. Okay, so I'm going to draw this webinar to a close, because I'm aware that there's quite a few people who also want to make it to the UniProt webinar that's starting at 4 o'clock. So thank you to Fen for presenting the webinar, and to Gustavo for helping out with the questions, and of course to all of you for joining us today as well. Thank you, and I hope to see you again in another webinar.